Alright, Unit 6, Oxidative Phosphorylation. So this unit was one of the more frustrating ones for me because there's so much in the unit that you don't need to know. There's a whole lot of information, so I'm going to simplify it to these main 8 topics that are the ones that are most of the test questions I've ever seen. So we're going to talk about the electron transport chain, reduction potentials, partial reduction and cleaning up the free radicals, antioxidants, the basics about ATP synthase, not very much about it, GPS and MAS shuttles, total ATP yields, and of course regulation. So a quick overview of where we are before we start getting into the electron transport chain. Remember, we started with one glucose molecule outside the cell. And it came in through the cell and went through glycolysis. In glycolysis we had a little ATP and NADH. The product of glycolysis was pyruvate, which then entered the mitochondria and through the PDH complex became acetyl-CoA. The acetyl-CoA entered the Krebs cycle, and you got a bunch of things out of that. You got some NADH, some FADH2, some GTP, and all of those are for energy. So the NADH and FADH2 specifically are going to come into the electron transport chain. We'll talk more about that. Next, the electron transport chain pumps out some protons into ATP synthase to power the release of ATP, and that's our end goal, making energy. Later on in the unit we're going to talk about the MAS and GPS shuttles, just enzymatic shuttles to get mainly that NADH into the mitochondria from the cytosol. So it's good to be able to write all of this out and be able to write out, all 10 steps of glycolysis, every part of the PDH complex, the 8 reactions of the Krebs cycle. Good practice for the test. So where we're going to pick up is the electron transport chain, which, like you've seen, he draws it into 4 separate complexes. Alright, I have the full names listed here. What he mainly refers to it is just by the Roman numeral. You don't need to be able to draw out the exact every single process of it, but the things to know about it are that NADH plus H plus is what enters. And as it enters, it's going to go through complex 1 in the box here, and in blue are the prosthetic groups. We have FMN and iron. Anytime you see iron, it's an iron-sulfur part of the enzyme. From the first part of the complex, you're going to get 4 protons out of it. So there's 2 entryways into the electron transport chain. Complex 1 deals with the NADH. The next complex, complex 2, deals with succinate, or FAD. That's the direct link, succinate to fumarate, to the Krebs cycle. So FAD and iron, remember iron sulfur, are the prosthetic groups. So you could say anytime you see this FMN or FAD that they're both riboflavin dependent. This one is not a proton pump. You get 0 protons out of it. That's going to come into play later. But if you notice, as you enter them, enter either 1 or 2, the same thing overall happens. Q picks up 2 hydrogens. Q picks up 2 hydrogens. So no matter which way you came in here, you end up with QH2, and that goes into complex 3. Complex 3 has irons in it. It has a total of 3 heme groups and an iron-sulfur part, and that pumps out 4 protons. passes them on to some more iron, which this part here now leads into complex 4. The same thing right here. Complex 4 has a couple coppers after the first iron. It has 2 heme groups, and the last part that you have to know is that there's a half oxygen molecule that turns into water. So let's see, to try to simplify this, the main things to know are what starts it, the prosthetic groups. Complex 1 is the largest, complex 2 is the smallest. Know that you get 4 protons, and then 2 protons out of complex 1, 3, and 4. And know that the last part was oxygen into water. This is going to come into play when we get to the ATP yield. Because you're missing out these 4 protons, you're going to miss out on more ATP if you enter in directly through the Krebs. So it's the physical link to the Krebs cycle is this reaction. We're going to talk about that a little bit later and how that relates. There's a chart in your notes that says reduction potentials. I put a question mark here because there's a lot in the chart, but I regularly see a question come up about the reduction potentials. So the ones that are most relevant to us are the top one on the list, going from alpha-ketoglutarate all the way to succinate plus CO2. And these are in order of highest and down to the lowest here. And the other ones that come up a lot are hydrogen into 2 protons. I would know that NADH and NADPH are very similar, and they are more than FADH2. But other than that, it's one question if you want to kind of familiarize yourself with the whole chart, so you can, if the question does come up, hopefully you'll be confident in the answer. This was the answer to our test, that NADH was the highest one out of everything he listed. So yeah, can't help you much on that one. If you want to memorize the whole chart, it might save you a question. Speaking of lots of memorizing, this is the partial partial reduction. Basically what's happening here is in the electron transport chain, when you have Q, instead of Q getting both hydrogens, sometimes it picks up just an electron. That free electron that binds right here is called a free radical. Free radicals like to bind to oxygen and cause problems in your body, so your body has enzymes in order to clean up these free radicals. So we're going to take this one step at a time. First thing that happens is the electron binds to Q, so you get this Q with the free radical. Anytime something's underlined in green, that's what's coming in next. So an oxygen that's floating around in the cell is going to take that electron. So now the oxygen has the negative charge. Q no longer has a negative charge. Anytime I put something in parentheses here, that means it's leaving the reaction. We no longer care about it. So now that we've taken care of Q, Q can go back up here. We just don't care about it anymore. No enzyme is required for that reaction. So we're going to wait until that happens twice. So we have 2 of these oxygen molecules. So once this will happen 2 times, and here's where we're at. two oxygen molecules with the free radical attached. Entering in two protons. So those two protons, two H's plus these two O's, we're going to do H2O2, or hydrogen peroxide. That'll take care of one of the oxygens, one of the two. The other one will go away. You're back to that oxygen. It's in parentheses. We no longer care about it. It's back to where it entered up in this step here. Two glutathiones enter here in their reduced form. GSH is the reduced form. So glutathione peroxidase neutralizes this hydrogen peroxide into water. And then you get, this is glutathione again down here, GSSG, in its oxidized form. And now we want to basically recycle that back into the reduced form. The reduced form is more useful for us. So we enter in NADPH plus H, and that is catalyzed by glutathione reductase. So when after that reaction takes place, you have the two reduced glutathiones again. That's in parentheses, we no longer care about it. It basically floats back in the cell up to here. But now in the process we took NADPH and it became NADP+. So we have a bunch of extra NADHs in the cell. So one of those is going to jump in and give its hydrogen to the NADPH. And you're left with a NAD+. So that's how I wrote it out, linear. If it makes more sense the way he has it in his notes, learn that. But these reactions will always come up on tests. Usually he'll show one full reaction, and he'll say what enzyme does this, or he'll ask things about the enzyme. Oh, sorry, the last enzyme was NNT, or nicotinamide nucleotide transhydrogenase. NNT I don't think is his proper abbreviation, I just made that one up because I didn't want to write this out a bunch of times when I was memorizing this. So a couple things to note. Superoxide SOD, I'm forgetting the full name of it, but it's in your notes. SOD can be in two forms, either in the mitochondria or the cytosol. And if it's in the mitochondria, it has manganese attached. If it's in the cytosol, it has copper and zinc. That could be an easy question right there. The glutathione peroxidase has selenium in it. Selenium attached to the cysteine. The last enzyme down here, NNT, is membrane bound. Membrane bound comes up quite a bit. And this reaction right here taking that extra hydrogen peroxide, I need to mark in that oxygen, but taking in that extra hydrogen peroxide and turning it into water plus oxygen can also be done by catalase. So you have two options. After these first steps happen and you get to hydrogen peroxide, you can either go the glutathione peroxidase if that's around. If that's not around, you have catalase, which is one of the fastest enzymes. It's a heme enzyme, and it takes that hydrogen peroxide, turning it into oxygen and water. A few other side notes. Glutathione is glycine, cysteine, and glutamate, and the cysteine is attached to glutamate's R group. I said this before, that GSSG is in its oxidized form and GSH is in its reduced form. I would memorize that table of antioxidants ATP synthase, there's not much that comes up about it. I would know that it has it's on the inner mitochondrial membrane, and the F0 is the proton channel part, and F1 is the catalytic part, or the one that makes the ATP. But all of those angles and all that, I didn't learn it. It never came up on questions. So know that the proton does not make the ATP, it simply releases the ATP. And it takes 4 protons to make one. Not really making one, more processing an ATP. 3 of the protons go to ATP synthase, and 1 of them goes to ATP ADP translocase. And that's to export it to wherever it needs to go. So because we know we need 4 protons per ATP, when you have 1 NADH makes 10 protons, and 1 succinate makes 6 protons, that's where the numbers are coming from for the ratio. 4 ATP per, and 1 NADH gets 10 protons. If you forget where that 10 protons number is coming from the electron transport chain. When an NADH comes in, you get 4 plus 4 plus 2. So that's 10 protons coming from the electron transport chain. If it comes in as FADH, which is through the succinate fumarate complex 2, you're skipping those first 4 protons. So you're losing that 1 ATP. So you enter in through here, and you get 4 plus 2, so you get 6 protons. That 6 protons is right here, and that's why it's 1 less ATP. Remember, this is for the mitochondria only. In glycolysis, there was some NADH that was made in the cytosol. And that cytosolic NADH can get in 2 ways. The first one is the GPS shuttle. So the GPS stands for glycerol 3-phosphate. You take an NADH plus H in the cytosol, and that binds with DHAP from glycolysis, turns into glycerol 3-phosphate. Glycerol 3-phosphate then passes off those extra hydrogens straight into complex 2. So that's complex 2. Remember, that one's going to get less ATP. We'll talk about that in a minute. The enzymes here, the first one is the cytosolic glycerol 3-phosphate dehydrogenase, and the other one attached to the mitochondrial membrane is the same enzyme, except it's called mitochondrial instead of cytosolic. The thing to know about this second enzyme here is that it's riboflavin dependent, has the FAD prosthetic group, and it's membrane bound. MAS, or malate aspartate shuttle, you're taking this cytosolic NADH plus H, and you're binding it with oxaloacetate. And those together are going to make malate. The enzyme for that is malate dehydrogenase, cytosolic, because it's on the cytosol side here. The malate can enter into the mitochondria, find an NAD+, pass off its hydrogen, its extra hydrogen, so it's going to turn back into oxaloacetate, and this NADH can go straight to the electron transport chain, the first complex. That's why it's going to get more. So you're going to have 10 protons rather than the 6, like from the GPS. Just remember, the MAS shuttle, MAS makes more ATP. Everything else is recycling. So the oxaloacetate is going to bind with the glutamate, or react with the glutamate, to make alpha-ketoglutarate and aspartate, which are both going to leave the cell to meet up here. They're going to react again. Oops, I'm off the screen. We're just going to come up here, react again, and form glutamate and oxaloacetate. So all of the enzymes here, number 1 and 2, are both malate dehydrogenase. The one on the cytosol is cytosolic, the one in the mitochondria has the M attached to it for mitochondria, and then you have the same sort of thing with reactions 3 and 4 here. They're both called either aspartate transaminase, mitochondrial or cytosolic, or aminotransferase is the more general term. So know these shuttles really well. They come up in a couple questions. The other thing to note inside and out is the ATP yield from a glucose. So in glycolysis we had a net of 2 ATP. Remember, we produced 4 ATP, but we invested the 2 at the very beginning, so minus the 2 we invest, there gets the net ATP of 2. We also make 2 NADH, which if it goes through the GPS shuttle, it enters complex 2, so it's going to come in as FAD and only get 3 ATP, whereas if it enters in the MAS shuttle, it's going to enter in complex 1 and get 5 ATP. It'll continue to be NADH. So complex 1, complex 2. The PDH produces 1 NADH, which is worth 2.5 ATP, but that happens twice. For every 1 glucose, there's going to be 2 PDH complexes that happen. So we're getting 5 ATP total out of the PDH from 1 glucose. From the Krebs cycle, you get 1 GTP, 3 NADH, and 1 FADH2. That's per 1 cycle around. So that's 10 ATP per cycle. But you're going to have 2 complete cycles per glucose. So per glucose you have 20 ATP coming from the Krebs cycle. So when you total everything up, if you're coming from the MAS shuttle, you're going to have 32, or you'll have 2 less if you're coming from the GPS. So make sure you know all that. Make sure you know that this is all cytosolic. So you have 2 cytosolic ATP out of the 32, so there might be a question that says that. How many cytosolic ATP are produced. So you do 2 divided by 32 equals about 6%. And he would have clarified using the MAS shuttle for that one to get the 32, or else it would be 2 divided by 30 for the GPS shuttle. All of these NADHs here and the FADH2 are eventually all made by the electron transport chain. So the electron transport chain is not producing any of these, but it's processing the ATP. So if you go through the MAS shuttle, you're going to have 10 NADHs, totaling 25 ATP. And you'll have 2 FADH2s totaling 3 ATP. That 10 comes from 3 times 2, so you have 6 coming here, 2 here, and 2 here. 2 plus 2 plus 6 is the 10 NADHs. If you're going through the GPS shuttle, these NADHs through the GPS shuttle start functioning as FADH2 because they're entering complex 2. So that's why you would get only 20 from the NADHs and 6 ATP from the FADH2. So these are the total amounts produced by the electron transport chain for either, and then you have to add the 4. The 4 are coming from 2 glycolysis ATP and 2 Krebs cycle GTP. The 4 coming from these two, plus this one that happens twice. So the regulation is just memorizing, there's no really easy way to do it. It's all inhibiting oxidative phosphorylation, so you don't have to memorize what's inhibiting and what's stimulating. So the different parts of oxidative phosphorylation starts with the electron transport chain, amytal, and these are names I can't even pronounce, so I'm not going to bother. The way I memorized this one was Amy is a rotten one. 1, 1. Complex 4 are some things that we know are being poisonous, like cyanide and carbon monoxide. There's also a nitrogen in there. I would know that ATP synthase is blocked by proton blockers, oligomyosin and DCC. I would know it can be uncoupled by DNP or UCP1, which is the same thing as thermogenin. These two block ATP export, and then just remember ATP itself slows down oxidative phosphorylation. If you have ATP, you don't need to keep making ATP. The very last slide lists the proton gradient's other functions. That usually comes up on a question, so I would know that slide really well, what the other functions of the proton gradient are, other than to release the ATP. So yeah, this is a tougher unit just because there's so much extra on the slides, but focus on these things, know these topics inside and out. That's what comes up the most. That's all I got.